We are coming back to chapter 25 now, which was all of the tissues. This was one of our first chapters that we covered, but we're going to talk more about muscles. Just a little recap about muscles. Of course, they are the motors for our body. It's what allows us to move. More specifically, inside the cells of our muscle cells, there are protein fibers called myofilaments, and they are made up of two main proteins called actin and myosin, and they make up these myofilaments that we'll be talking more about today. Now, there are three different types of muscles that we'd mentioned again back at the beginning. So smooth muscle, skeletal, and cardiac. Our summary table here shows these three types. So smooth muscles are called that because they do not have any striations. These are going to be on the walls of our blood vessels, the stomach, and the intestine. They allow for rhythmic and involuntary contractions. We're not thinking about these things. Skeletal muscle, those are the voluntary muscles. They are striated. We'll talk more about that. So again, things like walking, lifting, talking, all of those voluntary movements are provided by skeletal muscle. And then finally, our cardiac, as the name implies, these muscle cells are going to be found in the heart and allow the heart to contract. So a little bit about our smooth muscle. Again, these are long, kind of a spindle shape. They have a single nucleus. And these are the least organized of the three types. And again, as mentioned in the table, they're on the walls of the blood vessel and the guts. Now our skeletal muscle, these move our bones. So they're going to be attached to bones. We'll be talking about insertions. And that's where they're going into a bone to allow it to move. So they fuse into one very long fiber that have a lot of nuclei that are pushed on the periphery. So whenever you look at these, you'll see those small little nuclei on the edges. And each muscle fiber, again, has these myofibrils made up of the myofilaments that consist of actin and myosin. So this is really what we're going to be focusing on in this section. And again, these are just showing what these skeletal muscle fibers look like. So they have what are called myofibrils, which just look like little lines that go down to make up this tissue. Inside of the myofibrils, there are myofilaments, which are the little small bundles inside here. And each myofilament is then made up of the actin and myosin. So again, we'll be coming back and talking about these parts. Now the cardiac muscle, again, these are a chain of single cells, each that has its own nucleus. These are organized, they're branched and kind of interconnected to form a network. So these use a lot of gap junctions that allow some signaling and communication between the two, particularly electrical impulses to move from one cell to another. So a nerve can cause multiple cells or can communicate with multiple cells by communicating with one that then relays the message. Now, how do these muscles actually work? So again, we're going to focus mainly on our skeletal muscles. So first of all, there are tendons, and the tendons are connective tissue that actually attach the muscle to the bone, that allow the muscles to then move the bone, which is their job. So because there's this attachment, we have what is referred to as the origin. That's of the muscle at the end of the muscle that's attached to the bone. And this part remains stationary during the contraction, so it's not going to be moving. Now the insertion of the muscle, that's attached to a bone that moves whenever the muscle contracts. So the bones pivot about these flexible connections, of course, that are called our joints. So looking here at the muscular system, again, the origin of a muscle here. So if we look here, this is where this muscle is attaching to the bone. This is the stationary part. Then the insertion of this muscle to the lower part of the bone, that is going to be what's actually moving. So the origin remains stationary. The insertion is going to be moving throughout the contraction. We also talk about flexors and extensors. So muscles that can pull or only pull, again, because of those myofibrils, they're contracting rather than expanding. So the muscles of movable joints of vertebrates are attached to opposing pairs, and one is called a flexor and one is called the extensor. So whenever they're contracted, that's going to move the bone in different directions. Right, so if we look here, so essentially we have what are called muscle pairs, and a good example of this is seen in the leg. So the hamstring is a flexor, and the quads on the opposite side of our femur are the extensors. So again, these are moving in opposite directions. So flexion, we are decreasing this angle here at the knee. Extensors, we are increasing the angle or extending it. So in this case, whenever we bend the knee or bend the leg, our hamstring is flexing and the quads are extending. So these are muscle pairs that work in opposite directions to allow movement. Now, a couple different types of contractions that we'll talk about. Isotonic contractions, this is when the muscle shortens as it contracts. And then there's also isometric contractions, and this is actually whenever the muscle does not shorten, whenever it contracts. So this would be associated with trying to lift something really heavy. So the muscle isn't actually shortening, but again, there is a contraction happening in both of them. Isotonic, the muscle gets smaller. Isometric, it does not. So how these movements actually occur is referred to as the sliding filament model. So this describes how the actin and myosin actually work together to cause muscles to contract or shorten in the isotonic. Now the head of what's called a myosin filament, again, these are proteins, so it has a head region. It binds to an actin filament. So you have myosin attached to actin. So whenever the muscle contracts, the myosin head flexes, and it literally pulls the actin along that it's attached to. So again, that myosin head flexes and it pulls the actin along with it. Now, in order for this pulling to occur, we need energy, and that's provided by ATP. So that's used to release and then unflex the myosin head. So that allows it to return to normal. And so actually energy is not only needed for contraction, but it's needed to relax the muscle. So it's the energy from ATP that allows the myosin to release the actin and go back to normal. And so in rigor mortis, whenever a body is hardened after death, it's because there's not enough energy to release or relax the muscles, so they stay in a contracted state. So this is a better visual here of exactly what's happening. So again, we have the actin filament here. We have the myosin down here, which the myosin is thicker, actin's a little thinner. But what happens in contraction is the myosin head, this part of the protein, literally attaches to the actin. And then as it flexes, so again, that angle gets shorter. As it flexes, it literally pulls the actin along the myosin. So it slides across it. Again, energy, though, is needed in order to let go of the myosin. So here our energy is released in order to let go of that myosin head from the actin. Then once energy is expended, then it goes back to its normal state or the uncontracted state. And then if there's a stimulus, again, that myosin head bends, it slides the actin past it to allow contraction. Again, more energy would then be needed to release it to go back to the natural state. So the unit called a sarcomere here is what's referred to as the contractile unit of the muscle. So within this actin filament, they're actually anchored to one end of this unit called the sarcomere. And that anchoring end is called the Z-line. We'll see a picture here in a second. And then myosin is interspersed between pairs of actin filaments. and those can be connected on either end of the sarcomere. And what happens is as the actin filaments are pulled, again, by the myosin, so the head is attaching, pulling it along, then those Z lines are coming closer together and the sarcomere shortens, and that is cell contraction. Those Z lines coming closer as the myosin head pulls the actin closer together, it brings the Z line and shortens that sarcomere. Again, here's our picture just to show this. So here at rest in number 1, so here's our Z line on both sides. So this whole thing is a sarcomere from one Z line to the other. Again, we have our thin actin filaments. That's what's actually attached to the Z line. The myosin itself isn't attached to that Z line, only the actin. But then again, our myosin heads attach here to the actin. As they contract, it pulls those Z lines. So it's pulling the actin and it's shortening them. It's bringing those Z lines closer together as the myosin heads flex and pull the actin with it until it's completely shortened and our Z lines have come in. Again, this would be a contracted state. Now we would need ATP to be applied to release that myosin head and allow the actin to go back to normal. So essentially that contraction is when these Z lines are coming closer together, and that's occurring because the actin from both sides are coming closer as that myosin pulls it along or slides it along that myosin filament. Now, calcium is also really important for contraction. So in the relaxed muscle, there's access to actin by myosin. It's actually blocked by a protein called tropomyosin. So again, the reason that muscles don't stay contracted, they need some type of signal. Well, there's this kind of inhibitor called tropomyosin, so it prevents the myosin from attaching and pulling the actin. However, in the presence of calcium, that tropomyosin shifts away, and now actin and myosin can bind together. And these muscle fibers actually store calcium in a specialized part of the endoplasmic reticulum, the ER, called the sarcoplasmic reticulum. So we have the smooth and the rough ER for like protein production and our carbs and lipids. Well, there's a certain subsection of the smooth ER called the sarcoplasmic reticulum, and it stores calcium so that if a muscle was communicated with by a neuron to say it needs to contract, the calcium's already there, it releases it, and then it reabsorbs it and stores it back in that sarcoplasmic reticulum. But again, it's the nerves that stimulate that muscle. It causes the release of calcium from the sarcoplasmic reticulum. That triggers tropomyosin to be removed and allows actin and myosin to attach. All right, so in our picture here, it shows our actin and myosin filaments kind of at rest here. And what you can see is this tropomyosin that attaches to the actin. So now myosin can attach. So there's nowhere for the myosins to be able to come up and attach to it because tropomyosin is blocking all of those binding sites. However, whenever calcium is released, calcium binds to that tropomyosin and it causes it to move, and it opens up all of these myosin binding sites. So now that myosin can actually attach to the actin filament, and then that's going to allow, so our myosin now can bind to those open binding sites along the way. Again, it can pull the myosin along, causes that shortening. Again, ATP gets released and then, or gets added, and then the myosin is released from that binding site. And then the tropomyosin comes back and attaches, and then the whole thing can start over again. But we need that calcium to release the tropomyosin to allow the myosin to bind to the actin. All right, and that wraps up our muscle contraction.